Hello!
Hi there.
Welcome to Microbiology YouTube Journal Club, where we nerd out big about all things small.
My name is Danny, and in a previous life, I dropped out for my PhD in microbiology at the University of Chicago, where I was infecting skin I grew with MRSA. Nowadays, I'm a fact checker for pharmaceutical advertisers and the president of Biotech Without Borders, a nonprofit based in NYC dedicated to name the public with the tools of
My
biotech.
name is Faz, and in a previous life, I got my PhD in microbiology and from Imperial College, where amongst other things, I was making flesh-eating bacteria glow in the dark. And I've also since then worked as a research integrity specialist and as an editor for an academic journal.
Every week, we meet to talk about microbiology, and today it's our deep dive week where we look at each figure in a paper that we chose last week to learn and criticize the scientific findings. This is a journal club, and we encourage our audience to leave us questions and
Next week
comments.
will be our news week, where we survey a bunch of articles and choose an article for the next deep dive. So make sure to subscribe to satisfy your microbiology curiosities.
You can follow along with the papers that we discuss on either week in our shared Zotero library linked in the doobly
And we
doo
want to
below.
hear from you, so please use the comments or tweet us using the hashtag MicroTWJC hashtag. Uh,
So this week we have a malaria paper because I know you've been very excited about the malaria vaccine that's come out, and this seemed like a nice sort of middle ground where we could talk about malaria. That wasn't a clinical
yeah,
study.
absolutely right. The title of the paper is A Single Cell Atlas of Plasmodium falciparum Transmission Through the Mosquito. Falciparum. Uh, falciparum?
Falciparum? I'm not actually
I've
sure.
literally had
Yeah.
lectures pronounce it both ways, so I don't know what's right or wrong.
Yeah, so malaria disease, it's caused by a group of parasites and it affects a whole bunch of different vertebrates. And so I picked out some pictures here to show people that it's old. It's an old disease. There are reports, I was reading one of these review papers, and there are reports like, say, back to the like these cyclical fevers, right? Like
Right.
cycles of like fevers and chills, and that's characteristic of this sort of infection. And we'll see it like relates to the life cycle of the organism. So super old disease, and if you look at the this like phylogeny of it all,
Mm.
there's tons of different organisms, and they all have like a vertebrate host. So something with a backbone, with a bony backbone,
Yep.
that it causes the disease in. And then actually the other thing that doesn't show in this particular thing is they're all carried by mosquitoes.
Mm.
So it's this very specific type of parasite that mosquitoes suck it up in the blood of their of vertebrates, and then it grows in mosquitoes, and the mosquitoes inject it back into the same vertebrates that they got the blood from originally, and the cycle continues.
Yeah, exactly. And malaria is very, very different from anything we've talked about so far. So we talk about viruses or bacteria, but malaria are parasites. They're eukaryotic organisms. They're single celled, like almost our
Hmm.
cells. So they've got nuclei like us. They've got their own mitochondria. They've got a life cycle, and they actually go
Yep.
through sexual reproduction. So there are two malaria genders out there. I mean, sexes out there.
Yep.
Uh,
Yeah, it's crazy. Like if you think about that complexity, like when we think about bacteria, right? Like yeah, they have some sort of like they group in biofilms. They have some like social things, but this is way more complex than any. See that in their genome. You can see it in the fact that they live in so many distinct hosts, right? The fact that they're in the mosquito and they're in the and they're in a vertebrate, like those are very different environments, right? And I think that that can also speak to like the complexity of yeah, the life cycle.
Yeah. So going through the life cycle, the first stage it go well. We're going to start with because cycles. It's hard to say where it starts. But we're going to start with the mosquito biting the human. Injects these
Yep.
noodly versions of the parasite called I think sporozoites, and then those
Sporozoites.
sporozoites those migrate to the liver where they form these things called schizonts, which break out apart and release various I can't remember the name of them, but I think they're called so so we've got the life. Yeah,
Merozoites or gametocytes. This is like that's it's the entry their asexual stage.
yeah.
Um,
So
yeah.
this is where
So
yeah.
oh, go
Oh
ahead.
no, this is where they like kind of build up their numbers, almost building an army of them in your blood, and this is the part that's kind of where we get the symptoms and all the like bad stuff that happens with malaria. I think. Um,
Yes, that's right. Yeah, it's specifically this. I guess you can think of it as powering up. It's like this cycle of just cells exploding out, finding new red blood cells to get into, growing up, and exploding out again. And that cycle is, yeah, what contributes to this cyclical fever. And I guess the important thing also to keep, there's this huge diversity in all the different
Mm.
types of these Plasmodium. This is the general lifestyle for all of them, but different species may hang out at different stages, may be better at different stages, right? Like there's also this diversity in it, and that's why that cyclical fever is common between all malaria, but that's kind of all that is, right? Like there's some other symptoms and stuff that some malaria has, some doesn't, and
Mm.
immunity plays into that as
Yeah,
well.
and like,
Um, right.
yeah.
So maybe we should keep talking. So yeah. So that's the asexual cycle, that little circle that we drew. This is actually where the diagnosis of
Mm.
malaria comes from as well. That people take blood smears and they look at the blood and they say, "Can we see
Right, and
parasites?"
yeah,
And the red blood cells, they don't have nuclei, so like they don't stain for nuclei stains and stuff like that. And so here you can see like there's this like
yep,
DNA stain or something, and it'll like you can see dots. There's like a ring. There's different stages. There's a ring stage, I think, is a very common diagnostic measure. Like that's a very uncommon thing to
yep,
see inside of a red blood cell. Um, and so you can tell if people have malaria usually by a blood smear. And then I picked out an EM image just to show you like how many it's kind of
yeah,
how many like little merozoites are like growing inside of a red blood cell, and then that pops and infects a whole bunch of other red blood
yeah.
cells.
That's gross because you're almost seeing the merozoites moving under the skin of the red blood cell, which is kind of ugh.
Yeah.
And yeah,
Yeah.
and then after that they form gametocytes, which kind of have male or female kind of sexes, and those are what get taken up by the mosquito when it bites a human. And inside the
Yep.
mosquito, they can mate and they form a zygote, and then these
Yep.
briefly mobile organisms called ookinetes, which move to a specific area in the mosquito where they form oocysts that then burst out to form sporozoites, which migrate to the salivary gland, ready to be injected into the next human victim or mammalian victim.
Yep, absolutely. Yeah, and I that's like the creepiest picture of them emerging from the the midgut of the
Yeah,
mosquito. Um, so yeah, so this is crazy, right? Like this organism, I think seeing this whole cycle together really gives you the sense of how many body forms that this organism takes on during its life cycle, as well as how many different cell types it touches, right? It's intracellular, it's extracellular, it goes through epithelial membranes, right? It's going through a gut membrane, it gets into a gland, it goes from the dermis into the blood. There's all these, like, it has a very complex behavior. And, as mechanistic scientists, we're going to be asking questions like what genes govern those behaviors.
Yeah, yes, indeed. And also, I've moved on to the next slide prematurely, but I guess now's the right time to talk about
Yeah.
the actual mortality of it, because malaria is a scary disease for mortality. Um,
Yeah. It was, I mean, back in the day, there's no good records, right? Like
right.
we can't say from the ancient, from sort of prehistorical times how many people died, but there's a guess that it was quite high. And
Mm.
from the records we have here, I think there's a typo actually in this. The dates don't make sense to me. I bet it's supposed to be 1890 at the bottom there.
Right. Yeah, I think
um,
that's correct. Yeah.
Yeah. But so this is like 20th century, a graph of the 20th century, and malaria rates were quite high. This is annual deaths from malaria, and that's the worldwide, the dotted line with the diamonds.
Yeah.
So like the death rates were quite high, and there was a concerted effort around the world for people to
Mm.
eliminate this disease. And there are some new technologies that were out at that time that were proving really fruitful. They had
Mm-hmm.
DDT, which was like a great insecticide. They could spray it everywhere, destroy environments and ecosystems. But that's technology for you sometimes.
Yeah.
And then they also had a bunch of drugs were purified in the 20th century. Chloroquine, which is
Chloroquine.
a quinolone
Yeah.
drug.
Hydroxychloroquine, very famous drug. But yeah, it actually works for
Infamous,
malaria.
infamous. Yeah.
Yeah. Um, but
And
yeah,
so then in the 70s, it was basically the low. And places like Europe and North America essentially eradicated the disease
hmm,
because, like, if we think back to that life cycle, if you disrupt it, right? If there aren't people walking around with gametocytes in their blood, then there's no infection chain, right? Like
right,
you have to have those infected humans around. You have to have like a high mosquito population, and beating back the disease levels get lower. It's just harder to sort of to pop off, I guess, right, and maintain levels of disease.
hmm,
And so the big decrease actually came from, it was never that high, I guess, in Europe and North America, but into the 70s, it went super low. But the biggest decreases were actually seen in South Asia,
right.
in India. They made huge strides in getting the incidence of disease down. But then there was like there was drug resistant forms of malaria that kind of popped up. DDT wasn't so good to use. They didn't want to spread that everywhere. Um, Funding, I think, also was just difficult. Like, you spend a lot of time to eliminate disease and it's not 100% gone. I mean, this is like a scary parallel,
Well,
right?
yeah,
But 100% gone. And then people are like, do we really have to try anymore to get rid of it? And
yeah. This
so,
is getting into the socio-political kind of aspects of it, of kind of how we
mhm.
neglect the places like sub-Saharan Africa, and why that's kind of going to be an ongoing concern. Uh,
Yeah, yeah. I mean, I think that that's a really good point. Like, it was eliminated from like Europe and North America, and so the political will and sort of the global organization to continue pushing to eliminate it around the world kind of petered out.
yeah,
Um, and so in the 70s, so after the 70s, the all-time low, it just started rising again. And so like now we have the malaria deaths by region from Our World in Data sort of pick back up again. And similar in the graph on the left, it's the solid line with the circles. That's
hmm,
sub-Saharan Africa, and those rates just started to increase.
yeah.
um,
I mean, also I think there's like I mean, there's all sorts of things that affect. It's all very complicated, but I do think that there. I mean, because for malaria, I think we're getting about like it looks like about like 600,000 people dying like every year in these places, which is basically like
mhm.
having like half a coronavirus outbreak, but located in like just instead of the globe, just in sub-Saharan Africa and in Southeast Asia, but mostly in that area.
Right.
So I think we have to like kind of recognize how big that a deal that is and how big of a problem it is that we haven't solved it.
Yeah.
And now people are,
Yeah, and just because it's only happening in one
yeah,
part of the world, right? I think that that's one of those things that it's why, I mean, there's whole journals, right? They say neglected
yeah,
tropical diseases. And I think malaria fits into that category very, very well, both from, yeah, like it is a tropical disease, but like that neglected aspect that because it's not top of the mind for very rich industrialized nations, the technologies that have been mustered and the organizations to sort of address that have been lacking in
but I
some
mean on
ways.
the other hand, like with global warming, the actual like ranges of the mosquitoes that carry malaria are changing and could affect more industrialized
Yeah.
nations. So now that so so if that's what it takes for people to get to notice malaria again, that's kind of unfortunate and says a lot about the world
Hmm.
that we don't really want to say on this show particularly. I mean, but we're mostly a science
Hmm.
based, so we don't want to
Hmm.
say how terrible the world is. But uh,
But
yeah,
there's something there, right? Like I think that maybe it's just important to remember that like for all the great science that's done in the world, it still matters who chooses to apply it and where, and who like sends that science to the problems that mean something, right? Who determines what the meaningful problems are. And
yeah, I
maybe we can
think
leave it at
we
that,
can
right?
leave it at that. But yeah,
Yeah.
so going back to the complexity of the life cycle of the of Plasmodium, the malaria
Hmm.
bug, it
Yeah.
and yeah,
On A and B, I picked leaked out some images of like the way in which these organisms have become resistant to two classes of drugs. So
Yeah.
quinolones and artemisins are both drugs that have been effective in treating malaria, but malaria, the parasites have all sorts of interesting mutations now that they can escape that.
Yeah.
And of course, vaccines, our favorite, or maybe our recently top of mind
Mm.
intervention, is a popular choice to try to deploy against this. But think about how all the different life cycles it has. Finding the right target for a vaccine is difficult. Um,
Yeah, I mean for any drug, because firstly, they're eukaryotic life forms, which means they're quite similar to humans, so it's quite hard to find drugs they'll target things that are unique to the malaria,
yep.
and the processes that are unique to malaria are usually unique to one aspect of its life cycle. So that narrows the
Mm-hmm.
targets even more, and also the fact that it's constantly evolving. It's got sexual reproduction, which means that it can actually reassort its genes a lot quicker than we might but would be helpful for us.
Yeah, yeah, no, I think that's a huge thing going for malaria. Like, and this is maybe like sort of like a sidebar, but sex, the evolution of sex as a tool, right?
Mm.
Sex is this way that we can reassort genes, right,
Mm.
very efficiently and with huge amounts of diversity, and in the history of times,
Yeah.
right? And so like, it just goes to show that like while it is an expensive adaptation, right? Like it means that you have
Mm.
to have these two body plans. You have to figure out how to get those body plans together. And you could go hermaphrodite way, and then it doesn't matter. But uh, like that gives this huge wealth of diversity to draw from,
Mm.
and yeah, you can see the signature of it in malaria populations. I haven't pulled it. I think it's not one of these, but I think it's one of the previous slides had a reference, and it was all about the genetics. It's where I pulled the phylogeny from. That paper has just like a great discussion of how like the parasites can recombine in a host and come back out again, right? And that generation of variation.
Yeah. So all it takes is one person to evolve like a resistant form of Plasmodium, and then if enough bugs like hit them, then that could spread it quite wide. So it is, yeah. So I mean, yeah. Moving on to vaccines,
Mm-hmm.
though. Moving on to the good news. Um, but yeah.
Yeah.
So sorry.
No, you go.
Oh yeah, no, there are now two sporozoite targeting vaccines, and again, we've got the same sort of issues where we need to target antigens, but they are constantly shifting throughout this life cycle. So you have to think, where is it vulnerable? And I think the thing that people hold in on is the sporozoites, because this is the moment when it first enters. We want to hit it that
Mm-hmm.
the earliest part of its life cycle, and then that will be more likely to stop that part of the disease.
Yeah, totally. Yeah, that I think that it to me it sounds like a really good stage to target it at.
Mm,
And I think also the trouble has just been like the difference between these two vaccines. So the first one was only 56% effective. This new one, which we were getting excited about right in our news
mm,
articles, is like has been shown to be 75% effective. It's the same it's a very similar antigen that they're using,
mm.
but the difference is just in the boost, in the way that they in the adjuvant, the sort
Yeah,
of the way that they tickle the immune system to recognize that antigen. And so I think it also just shows that getting the immune system to do the right thing once it is exposed to the right antigen is also one of the tricky parts about malarial immunity.
yeah, I think it's a big part of like all vaccinology is finding a way to communicate with the immune system to tell it that okay, this antigen we're showing you is something you need to be worried about into the future, and you need to respond
Mm-hmm.
in this way. But we're still trying to learn to understand the language of the immune system to be able to communicate that properly. And so
Yeah,
these like so we don't talk about adjuvants very much. We often focus on the main antigen, but adjuvants are really important in trying to figure out how to get the immune system to respond in the right way.
totally. So yeah, so pretty good news. Then like we have these tools, but of course like they're not 100% guaranteed, and it's
Mm.
always good to have more tools in the toolbox. And so research is still continuing on the malarial parasite.
Yes, for sure. Uh, and now we're getting on to the, the the topic of the paper: single cell RNA sequencing. Uh,
Mm-hmm.
Transcriptomics are a big thing. Being able to isolate individual cells to find out what the transcriptome is doing can tell you so much about what proteins are being made, how it's functioning, and in order to. But since you're looking at all these individual cells or the different individual situations, we have to use really advanced data analysis to try and understand what's going on from the perspective of the whole infection.
Yeah, totally. And going back to this fact that the life cycle is so many different life right, has so many
Hmm.
different stages, you can expect that if you sequence cells from all those different stages, they're going to be expressing different things at every stage. And that's like quite a complex data set that you wouldn't necessarily just ground up a whole mosquito, right? And like looked at all of the transcripts that come out that go to
Hmm.
Plasmodium, because when you grind up a whole mosquito, it's like an average, right? It's an
Hmm.
average of everything that's in there. When you just take out the blood, you get an average of all those things. And so single cell is poised to be very useful in this context. It's useful in also so many different scientific contexts, because of this, right? Because instead of looking at this average, you're looking at these individual data points, and that gives you better resolution on logical phenomena, right? Like
Yeah.
biology happens at the level of the cell in many cases. Having single cell resolution is like a really great tool to see complexity at that level.
Yeah, I mean, it's like if you try to like grind up human society and find the average human being, but you get a kind of a horrible mass of different parts, and you wouldn't know how it works. You just get an idea of the average. Whereas this is
Yeah.
more like isolating each individual person to find out what their job and their function in the society is, and then building a picture from
Yeah.
that. So
Totally. Do you know this? There's a bit of a sidebar. I want to say because people talk about an analogy of how does a biochemist versus a geneticist figure out how a car factory works. Have you heard that one
mm,
before, Fuzz?
I have not heard this one. This is a new analogy on me. Let's hear it.
Okay, so like the idea is like so a geneticist looks at the factory, right? And to understand how it works, they go in there and they take one of the workers on the line. They don't know what they do, right? They just like they remove the worker from the line, and then they see the cars that come out. Okay, like based on the fact that these cars don't have steering wheels anymore, the person I removed, right, is the person who puts the steering wheels on the car,
Mm,
right? And in this analogy, it's like these people are the genes that are working in the organism to make something happen. And so the biochemical approach, right, says that like they go in and they catalog all the things that they see inside the factory, right, and then they try to put them back together to see if they will make the car, right. And so they'll take like, oh, they put these steering wheels and they try to mash them together and say, oh, here we go, like I've made a car. And
mm,
so taking that analogy of like a living organism, like a factory, right, where the people are like
mm.
genes and like all the little objects are like things inside the inventory, single cell sequencing, right, is letting us go in there and really pick out like individual
Mm.
elements rather than averaging everything together. So like what if every individual was a cell in an
Right.
organism, right? So like not from a cell perspective now, the factory is an organism. If you just grind up that whole factory, right, and you say, okay, like this is the inventory, right, it doesn't give you the sense of those individual roles that are being played by the actors, and so that's what single cell sequencing I think is giving us. It's giving us more specificity to the roles of cells.
Right. So that brings us, I think, to the first experiment of this paper, which is in Fig 1A and Fig S1, where they kind of
Yeah.
do the. So the thing that the unique USP of this paper is they're not just looking. Usually you look at like human blood, but they're looking at the mosquitoes. So trying to
Mm-hmm.
isolate the part of the life cycle that's happening in the mosquitoes, because that doesn't necessarily have the same level of scrutiny as the rest of the life cycle, because it
Yep.
doesn't affect us humans. Again, like we're research is
and the
dependent
and
on,
the
yeah.
and the other important thing about this one is that they're looking at this Plasmodium falciparum parasite. And previously, when they did the cell atlas, they were looking at a different Plasmodium species.
Yes.
So like they're also finding one that this is like one of the more deadly human pathogens. So like we're hoping that there's also going to be more relevance to the data that they're pulling out.
Ah, yes, yes. The previous one was Plasmodium burgeri, which infects mice. So it
Mm-hmm.
tends to be the model because you can see the life cycle in mice a lot easier than we can in humans. Uh,
Yeah.
because it's a lot harder to do that. We get the consent for the kind of experiments we need to do. So usually we do something. You have a model on mice to get the framework, to get an idea of the general aspects of the life cycle, and then from that we can build
Mm-hmm.
up on that to take whatever we can pick out from uh, the the thing that infects real humans, and then see what the similarities are, what the
Yeah,
differences are, and make deductions based on that. So
totally. This was the first run, was the model, basically the model Plasmodium. But this is a data set that they're going to make with one of the more relevant human pathogens.
yeah, so they screen using PF25, which is a biomarker for female gametes, and DRAC5, which is just a marker for nucleic acid. I think for the nucleus. Uh,
Yep.
yeah.
Yeah, and and this particular sample is being taken from the blood meal.
Mm,
So ideally, using this screening strategy, they should be getting some mixture of the activated gametocytes, things, the zygotes, this like kind of like pre-form called a retort,
yep.
and the kinates. So that's what is in this particular sample that they've purified. So they're able to sort out these Plasmodium cells.
Yeah, and so next we're focusing a bit more. So again, we've got uh,
So in this one, they want the asexual stage,
right.
which happens in blood. The way that they get those is all cell culture. So they just have like a flask that's full of red blood cells that they have some plasmodium in. And I was reading about how they induce gametocytes. They just kind of wait.
Yeah.
is the induction. Yeah.
Yeah.
So I think the idea is that, I mean, they have their own program. They have their own genetic programming, right? These parasites. And so if they get to a certain concentration inside of culture, then they start bursting out and becoming gametocytes, because that's, I guess similar to what they would do in our
They
bodies.
still have that clock, even if they're not inside the human body. They still have that internal timer for the cycling.
Yeah.
So yeah, you have to wait for I think 15 to 17 days for it to burst. And
Yes, yes. And so they use two strains Plasmodium here. They purified out the gametocytes using DNA stain, use a host.
I don't know.
And an RNA stain, pyronin.
Yeah.
Yeah. And then they can assign the sex to these gametocytes using PFS 25, which is the female chain, and the dynin heavy chain for the males.
Right. And yeah.
So yeah, I don't know if we want to explain this well a lot before. And yeah, it shows that they can see the two groups of them. Oh, I guess there are some that don't stain for either.
Yeah, so
immature. Yeah,
they're immature, so they're not quite ready to be out on their sex. But,
and so I think that's I think that's probably, you can think of that as an artifact of the way that they made their gametocytes. Right? Like there's no like magical signal that they use to make them happen. This is part of the natural progression. And so not everything is on the same time scale, right? I think that's what's being shown.
Yeah,
there.
So a lot of this paper, they're not going to be infecting humans to get the information. They are reproducing the kind of stages of the life cycle in petri dishes or in various kind of cell cultures that are designed to simulate. Oh, but they do it in mosquitoes. We are going to get to that.
And mosquitoes.
Yeah, so that brings us to Figure 1A plus S3, where they take infected mosquito midgut. So they feed these mosquitoes the malaria, and then afterwards they leave them for a while, and then they dissect out their midguts or salivary glands, and they have to time it out because they have to wait for the actual parasite to go to the right place. But
yeah, like the earlier time point is when they were expected in the gut. So like at the earlier time point, 12 days is when they take the gut, but then 17 to 20 days is when they would expect it to have migrated to the salivary glands. So then that's when they take the salivary glands. And then they even take like an in-between time when they take the hemolymph or the blood of the mosquito at that point. And so yeah, they're really capturing these these are sporozoites too, right? Yeah, like I think once they burst from the oocysts, they're sporozoites. So like they're capturing these sporozoites from all these different stages. Oh yeah, and then the last one. They also then have mosquitoes that were infected, and then they make them feed on some just like a mixture of sucrose and serum,
Mm.
and like mosquitoes spit a little bit when they stab something,
Yeah.
and so they essentially they also capture the sporozoites that come out from the
Yeah.
spit.
So that's how they get. I mean, that's how you get the sporozoites that just been injected. So yeah.
Hmm.
Um.
Hmm.
Yeah. And I think.
So, from all of those different types of protocols to purify out cells, they do single cell RNA-seq on them. And so we've discussed this in detail before, is that there's a method for separating cells into their own little bubble, and then those bubbles are lysed, and every single RNA in there is sequenced, right? This is the magic of our
Yeah.
omics universe these days. And this table and figure S4 is just showing us some of the cleaning procedures that they did to the data to make sure that they're getting only high quality reads. And so one of the things that they do is they cut off the number of genes that are being expressed per cell. So you can imagine if a cell's only expressing like 5 genes, but on average, most of the cells are expressing 500 genes, then something's gone wrong in that particular action, and you don't want that data to influence the rest of the set. So
Yeah.
they cut things off.
Yeah. They
And
could
they
be.
chose a different cutoff for the different cell types because different cell types actually have different expression, amounts of expression that they do.
Yeah, definitely. So you can see like a lot for I think these are the sporozoites. They have like various peaks, and then there's got a small
Hmm.
peak which would be kind of various things. So sometimes they can be created by the technical issue, the technique. So
Hmm.
primerdimers or other chemical things. It could be the dead cells. So there is a concern about.
Yep.
So I mean, I know for when they're looking at gametes, they're concerned about gametes that don't mate or live too long, and then they end up kind of dying off and leaving a lot of kind of because the transcriptome, you want to find ones of healthy cells that are performing their life cycle.
Yes.
Dead cells can skew your analyses, and you want to, and
Hmm.
the things we want to treat against are the living cells. We want to, we want to isolate all the useless stuff from it.
Yeah, yeah, so absolutely. Yeah, that's really good pointing out in figure C there, right? Yeah, they're essentially removing that bottom peak.
Yeah,
So there's still variation in their sample, right? But they're eliminating the part of the variation that is likely we want to study. And then they take all the genes from those cells and they're all matched to a single cell and they normalize them just to smooth out the data. They normalize it against a log
right.
scale.
Uh, and then they go put through the UMAP kind of data analysis. Um,
Which we've also talked about before, and maybe won't say too much in detail here, but essentially, that's a huge list of genes that come out, and they want to show that in a graph. You have to reduce the amount of dimensions, right? Every gene
mm,
is a dimension that could be right anywhere from like zero expression to like 100% expression. Imagine, right? And so you can't draw a graph that has hundreds of different axes, right, where that go from 0 to
right.
100. So they use an algorithm to preserve the similarity between different sections of all of that data, but collapse it down into 2 dimensions.
So almost as like what you're seeing here is a map of the gene expressions, and the closer points are, the more similar they are. And so
Hmm.
you can see here we've
Yeah.
got like the asexual kind of stages are all grouped together in like one group. The gamete stages grouped together. The ookinete sites grouped together, and this big
Hmm.
swatch here is the sporozoites, and that's divided down into the environment they're in throughout the because sporozoites have to be but live in mosquitoes and humans and in different organs within those areas. So you've
Yes.
got the sporozoites that are just newly born, and then they turn it into hemolymph sporozoites, which are in slightly dark shade of green, which are migrating through the blood. Then you've got the was it the salivary gland
Yep.
sporozoites, which live in the saliva, and then the activated ones, which are just the ones that have just been injected. Well, you got the injected ones, and then activated ones are the ones that are living in the blood.
Yes.
So yeah, we've got the whole like life cycle in one image and all the
Y-
diversity.
Yep. And they can even so that's that so those are all the dots and those different cell types. but they can also just look at the gene expression data and use an algorithm to cluster the gene expressions that are similar to one another. And so that gives us another thing to lay over the data. And you can see that it generally lays over in a very similar fashion, right? Like there are specific clusters that are mapped onto the same dots, right? That have been seen and that are mapped onto cell stage life cycle. And that's just, I guess what this is you can think of them as programs, I guess, right? That these clusters of co-regulated genes are programs, genetic programs that are running. And it's good to see that they map onto the different cell types, because you would expect that like the programs that are running should be different in different cell types.
Yeah, and you can even pick out small clusters, so you can see like what these different cell types might be doing differently. So, for instance, asexual has C6 and C2. The gametocytes, they should be clustering differently because they're both male and female in there, so C7, C4. And sporozoites, they're clustering as well slightly differently because, again, they've got differences throughout their life cycle. So,
yes.
And of course, like and the injected sporozoites might not be so different from the activated sporozoites that we find in this experiment, because there's a lot of interesting things that we can. There is so much data here that they've had to simplify it, and now I think the rest of the paper we're going to be pulling out bits of the interesting parts that they find here.
Yeah, I think that what they want to end up showing by the end of Figure 1 is that the life stage cycles and the clusters can be used sort of together to understand stuff about the biology, right? With certain life cycle stages, like that's telling us something about all the genes in that cluster as it relates to that life cycle stage. So yeah, let's see what we got next.
Yeah, we've got focusing on the different life stages. So we've got the asexual life stage, we've got the sporozoite, and we've got the ookinete stage. Yeah, so
these three stages, they just subset. This is basically a subset of the data, so that they can run this funny algorithm. Oh, wait, is this the one? Oh, no. This is just, in this one, they're just, oh, no, they're looking at over time, right? They're adding, they're running this funny algorithm called pseudo time.
Yeah, pseudo time. So this is an interesting one. The way I understand pseudo time is basically, okay, so let's say you want to find out what I'm going to look like in several years' time, but you don't want to look at you don't want to stay around for 70 years to see what I look like when I age. So instead, you look at my family, look at people, my parents, my brothers, my cousins, all those, and then from that you can kind of get an idea of what my life will be like based on my relatives. It's kind of the similar thing for parasites.
Oh yeah, I didn't think that's an interesting way to think about it. Yeah, because in their data set, if you think about captured from all these different time points, right? So they have all that information, right? They have all those different future forms. And they can look at them all together and try to order them in a way that makes sense. So maybe in your analogy, genes would be like the features on your face and stuff like that.
And like even when they're taking out, say, a mosquito at one time point, so the different parasites will be at different stages of the life cycle just because of variation. So using some clever mathematics, they're able to kind of try and predict what stage of the life cycle each parasite was, and then produce these kind of swishes that show the progression from one stage to another.
I don't think the input wouldn't say anything. What's good about this method is it's purely mathematical, right? It doesn't say anything about the life stages of the cells. But what it's seeing is the transcription data. So it's saying based on these different transcription datas, you can these cells over here are in between these other cells, right? It's building this like a spectrum. Right. That shows how the transcription is changing over how it just evolves, and it assumes a direction of some sort, right? You just choose your starting point and say, what's I guess actually you wouldn't know what the direction is. There must be something in the algorithm that gives it the directionality. But it's essentially building this kind of branching structure that says, here's the paths that you could go forward, right? Based on the snapshot of all these different things. And the success, if that algorithm is successful, they're able to draw a line essentially, and and map out like different stages. And so what is encouraging to see is that those stages then should map out well, or they're running them, they split out the different life cycle stages here, right? So it's saying within a life cycle stage, there is some directionality to the way that those cells are evolving, meaning that we're seeing a developmental program of these specific life stages. So it's like it's more zoomed in.
Yeah.
Right? We already know the life cycle stage is zoomed out. We don't need to have that proven to us. But what we do want to see in this data is, can it tell us about inside those life stages, like some of the more nuanced things that are changing? And because we do get good pseudotime representations, I guess, these arrows at the bottom, and that it seems to be going through the data set in a way that covers the whole data set, that I think is giving us confidence to say, yes, we can learn something developmentally from this data set.
That brings us to Fig 1D, which is using k nearest neighbors to determine the expression patterns across the life cycle. So
yeah. So this is like now they've thrown out the UMAP from the window, and they're gonna reapproach this with another clustering strategy that works with k nearest neighbors. So it's graph based. Things are supposed to be things that are close together again should be at the same. But I mean, you can see this graph. It doesn't work 100% the same as you would expect. There's some disconnect between the way that this is visualized and the way that the cluster is being formed.
But yeah, I mean, k nearest neighbors is essentially like trying to find like the things that are like kind of co-expressed with each other. So things that are points that are very similar to each other, like UMAP, but the display is kind of different. All the subtleties are quite different between these algorithms, and I'm not quite sure of them. But I think the main thing is like if there's things that are near to each other, they're very similar, and that's kind of how you interpret most of these graphs.
Yeah. I mean, this one's a weird one because I actually don't understand this particular element, like why they switched, because I think UMAP has some k nearest neighbors element in it. So like there's obviously something I don't understand, like that and there's a lot more clusters here than they built with the other method in 1C, I think. Right? And so there's just something, I think this method gives them better resolution on clusters. And it may also take into account velocity of gene changes is really similar, right? So like if two things are going up at a really similar time, then they're being grouped together. And again, like where the dots appear on this graph isn't the same as the cluster analysis that was run. It's just the best way in which that we could see the separation in dots.
Yeah, I mean that because these are visualizations that are trying to reflect something in reality. Sometimes using different visualizations can give us different perspectives. So rather than having something that'll group things by life cycle, you do want to see what genes are operating together. So if you get a gene that operates like at different points in life cycle, and there are other genes that are working similarly, I mean, we don't. There's so much about the malaria genome that we don't necessarily know. So actually seeing something that's co-expressed with the gene we do know about can actually tell us about what that unknown gene is doing.
Yes, absolutely. Yeah, so that's a really good point, that it's good that this analysis doesn't use the same visualization strategy that split things up by cell stage, because in some ways now we're getting at the more we're getting more granular in the analysis, right? We just want to know at the gene level what's going on, and then we can maybe try to map it back to cell type. I think that they will be doing that as we continue through this paper.
So this brings us to these gene clusters that they've been looking at and what they're related to. So again, like you said, they're more granular. So in previous one we see G1 relates to, say, the nucleus, G2 microtubule. So they might be doing different things throughout the life cycle, but it's quite interesting to know if they're doing different things as well.
Yeah, so the top go term, that's just what the most frequent thing is. It doesn't mean that all the genes in that cluster are doing that, but it's giving us that sense that like many of the genes are doing this type of function. Seeing the authors, I think, they're trying to justify the use of these clusters, right, as useful analytical units. And I think what seeing a top Go term emerge is telling them. And also the other important thing is that these align with those life stages, right? So that the bar, the colored bar above all the G's, right? That's the colored bar that aligns to 1A, right? All those different life cycle stages. And seeing that these clusters sort of map onto some of those life cycle stages, that also gives them like, oh wow, like, you know, this might make a lot of sense because in a certain life cycle stage, all those genes to see them regulated together, and that is what they're seeing here. So, you know, this is adding to their sense that okay, our analysis is looking good.
Yeah, I mean, again, this gives us so previous one with the UMAP, you can see the differentiation within life stages, but now we can go even a little further and make some guesses about the gene networks that are activated between those lifestyle stages.
Yeah, so if you remember in the UMAP stage, they used this clustering algorithm on it, and so that's what they're showing on the other side.
Oh, sorry.
Yeah, C.
Oh
That's
yeah,
fine.
sorry.
In 1C, we
Yep.
went there. They did a clustering algorithm, and then on B, they're doing the life cycle
Yes.
stages. So if we go back to their other clusters now, their G1 through 15 clusters. Essentially, they want to map that back onto B and C and figure. So E maps it back onto B, and then S7 maps it onto C. And so, again, those clusters roughly correlate to the life cycle stages. And you can see that in each gene cluster, the gene clusters now, those also correlate, right, a little bit, but some of them correlate more specifically with a stage, and some of them is more like all over the place, right? And you see the same thing in E, right? It's the same. It's almost like two ways of showing the same data, but it's actually two different analyses.
So now figure S8, which is looking at mutant screens of
So that they're going to pull in the first time of many times that they're going to pull in data from another source, right? And overlay it on their atlas. And like this is what's great about cell atlas research, right? Is that it's telling us things that it's very much in conversation with the literature, and because it looks sort of like in this wide way, you can see how well it aligns with things that we already know, and maybe even find gaps, right? Because science is just about asking very specific questions and getting that answer. But when you take these approaches that use big data, if you do a lot of work, which they do in this paper, to align that with published results, you can start seeing like, oh, some little gaps where you wouldn't have expected, right? Like not knowing information. New insights come out because you've compared things in a way that haven't been compared before.
Yeah, and this time they're looking at essentiality genes. So in previous work, what they did was they created these massive mutation screens where they'd give like malaria some crisp bars that will chew through their genome, create a random library of mutants, and then see which ones survived. So again, this is going back to the car factory where they're breaking different parts of the life cycle and saying, oh well, if this part breaks and nothing happens, you didn't need that part to make the car. But and the thing about all those like kind of screens where they were focused. First one was focused in blood. The other one was focused on a mouse model where they injected the schizons. So they were basically doing the asexual part of the life cycle, and so they classified the ones there as essential genes. But here they found a lot more. But the thing about this, the way this data is different, is that this one looks at the mosquito aspects of things that the other studies didn't. So
absolutely.
So I suspect that a lot of those genes that were described as inessential were actually essential, but for a different life cycle space that they weren't necessarily looking at previously.
Yeah, that's a great way of explaining it. So these are the same. So this is that weird clustering that they use to get the G clusters, but based on this data that came from either growth rate or from whether or not they even lived or died, right? If the mutation was done. And I think that what they're just at the end of the day, what they're trying to show is, yeah, you can see that that there are genes that are very in certain clusters there are more essential genes than others, I guess, but actually it's kind of well distributed all over everything. Yeah, I don't remember. I feel like they actually did have like a bit of a conclusion
Well,
that came out of this,
I
but
think the
yeah.
conclusion
Well,
was that
let's.
the genes that were expressed like almost in the salivary or like gametocyte kind of life cycles were not described as essential in those previous studies, which
Mm-hmm.
is
Mm-hmm.
kind of it's understandable if you understand the baseline of those studies, but it's also interesting and it also kind of highlights
Yeah,
the utility of this research, why it's important to look at this other side of the life cycle.
totally. All right, let's see what's
Next,
next.
right, so this is looking into the G1 cluster.
Yeah,
Um,
yeah. So you might have noticed in the earlier figures that that G1 cluster didn't seem to go with any of the specific life
yeah.
stages. Yeah, it's so diffuse along everything. And then by the clusters, G1 is in the top left.
Oh,
Oh,
sorry. Yeah,
no.
yeah. The cluster gene. Yeah, but you can see like in that
Yeah,
graph,
right?
it's showing the same thing. That I
Mm-hmm.
mean, the top go term was nucleus, but that's kind of vague. There's lots of stuff that happens related to that. So this is a question they want to delve into deep because if it's expressed like in all stages, then that could be a good target for things.
Mm-hmm.
Um, so yeah, and
And so
they. Oh,
they end up finding that it's a transcription factors that are
hmm,
in this group, right? These AP2 transcription factors and or APT2, I think, transcription factors. And this is known to work with chromatin remodeling proteins. And so one of the hypotheses that thing, right, is like, oh, maybe this is some sort of like regulatory cluster, right? And
hmm.
that's why we're seeing it pop up in all the different life cycle stages because every stage is going to need regulation of some sort. Um,
Yeah, and so BDP1 is called I think bromodomain protein, and it's
mm,
sort of related to translational histone modifications. So the idea that there might be some epigenetics happening in this stage of the life
mhm.
cycle, which is again not something I'm an expert on, but is an interesting buzzword. So um,
I mean, it's a huge field, right? Like, I think that
yeah,
that's the story of genetics is sort of is discovering all these different ways that modulate our genes. Like at the beginning, I'm not that any geneticist at the beginning thought that this was the case, but you'd think that once you get the inventory of everything, you just know how it works, but that's not the case. There are so many other little details about how those genes are used that that tell us what that expression really means in the context of a living organism.
yeah, so you've got these like heat maps of like that show DNA motifs, and I think at the moment I'm not sure I'm able to interpret these, but I think given more time, but it does like pull out like the kind of
This is
sorry.
it's I think what these two figures are is the best way to think of it is it's an expansion of the figure that we saw in 1E,
Right.
right? And they just instead of they just genes that they think are interesting in cluster G1,
Right.
and they're saying how do these all map out against the different life cycle stages.
Oh.
And you can see that there are so like in see that there are clusters of large dots, I guess, right?
Mm-hmm.
Uh, with different life cycle states. And so that would mean that different transcription factors in different life cycle stages. And that's the same thing in the heat map. You're seeing clusters of white, uh, different transcription factors, all part of this gene cluster, right? For different life cycle stages.
Right. I see.
Yeah. Oh,
Okay.
actually, well, this is not life cycle stages now. This is
Oh,
different transcription factors in gene clusters.
right. Yeah. So they're showing slightly different things, but it's kind of honing in on that knowledge of the because transcription factors are so important. I don't think we've really talked about them too much, but they are a major thing
Mm.
in all like kind of they regulate what they are the brains of what of the cells. They decide what the cell does, and
Mm-hmm.
so
Mm-hmm.
being able to pick these out can be quite fruitful for future research. Anyway, so moving on to figure S10, which is looking at unannotated genes. So these are genes that are almost entirely new that might
Mm-hmm.
that we don't know what they do. So
Yeah, yeah. And yet they are in. So wait, did you skip a figure in the last one?
I think I'm
Oh no.
no I. I
Oh
didn't.
no, you didn't.
I skipped
And
ahead
then you just
actually.
went,
Yeah,
yeah,
I skipped ahead and then I
right,
skipped
right.
back. So I went straight to S11. Then I'm back here to S10.
So S10, because I guess they want to argue for the utility of this approach.
Mm,
And so just the fact that there are a bunch of unknown things that are in the that are co-expressed together just means that these are open questions.
mm.
And they want to say that this could be the genes that are important between chromatin remodeling and APT2 just because based on like the story I guess that was told before, but like we actually have no idea
No,
what this would mean.
but this
Yeah.
is like kind of almost like what's it? It's evidence. It's kind of that coincidence. They're at the crime scene for all these things, so we're assuming they're involved.
Yes. That's right. And it would take a follow-up study to knock these out, to I don't know, purify some of these genes to really get at like what they are doing potentially in this
Yeah.
role. Yeah.
So that brings us to figure S11AB. So orthologs. So orthologs are genes in different
Oh,
species that
yeah.
evolved from a common ancestral gene. So like, because we don't use the term homologous because there are homologs that do different functions. So orthologs
Mm-hmm.
are ancestral genes that perform the same function.
Mm-hmm. And they have all this data. They have data from Plasmodium burgei, right?
Mm.
Because that's what was in the malaria atlas before. So like they want to be able to use those two pieces of data together because things that are conserved between the two, right, speak to common function the parasites, and things that aren't conserved between would speak to unique stuff. And so this is another way of teasing out more information from their data set is against a previous data set. And
Yeah.
so when they look at the number of orthologs, they see that there are some gene clusters that have more orthologs than others.
Yeah, and I think that's important for understanding because like a lot of our understanding of malaria's genomes comes from model organisms like burgei. So understanding
Mm-hmm.
the kind of commonalities are very important, and also understanding the things that are very different, so we can kind of extrapolate the right and things from these models. And
Yeah, and like close the gap, I guess, on the whole story of area.
yeah, and I think it tells us some interesting things about like the evolution of this. I was about to say bacteria, but this parasite. Um,
Yeah. So the figures, the heat map sort of figures that have black in them, that's just the color is coming from the homologs or from the
right.
orthologs, and the black is the unique stuff. And then they're just looking at different. So these are like families of genes that I don't remember. They have some significance in malaria study, but I don't know what it is.
But yeah,
All that to say, there are things. Oh, do you
oh
remember?
no, I was
Or
about to try and move on to the point, but because I don't
yeah.
remember the gene families. But what I do want to draw attention to is that these three kind of first parts where most of the homology is the areas that are happening in the mosquito. So
Hmm.
and a lot of the place where there's variation is the stuff that's happening in the mammalian host. So the
Yes.
sporozoites are where a lot of the variance is happening between the species. Whereas the
Hmm.
stuff that's very similar is the stuff that's happening in the mosquito.
Yes. And if you recall, I was saying that by the end of figure one, which is like they're still getting through
Mm,
it, right? Like by the end of figure one, they want to be able to say that the gene clusters that they assigned, right, with that k nearest
mm.
neighbors graph, like that those gene clusters are somehow significant, that they have functions that they're in of themselves, and this is one of the ways that they can show that, right?
Mm.
Just as you say, because we know something about this life cycle, we can take that knowledge and we can add that as construct gene cluster thing that happened. But in doing so, then it means that everything in that gene cluster that we don't understand, right? Those genes that as in the previous slide, right? Like there's a whole things we don't understand. We can start to say, well, maybe they have something to do with, right? All this context that we're adding in there.
Right. Yeah. I think we can move on to the
Okay.
next slide, which is
Hmm.
11 CDE, which is looking at amino acid conservation. So that's
Hmm.
amino acid sequences are conserved to maintain the structure and function of a protein or domain. So again, that's another way of looking at that orthology that they were talking about
Yeah, it's like
previously.
a more granular form of the orthology. And it's the same pattern that you were describing, but actually they even see a rather clear signal, which I mean, that's actually very hard to see in these graphs, but
Mm,
that G2 and G13
right.
are the lowest conservations, and then G13 is the highest FST score, which is a sort of another difference. It's a index, a type of difference
Oh, right.
index,
So,
fixation
like, the
index.
purple is the low conservation score. So, obviously, GS2 is the most hardest to read because it's the same color as a put. Because from my
Yes,
perspective.
yes.
But,
Yeah, and maybe it's easier to see in D, which is that same data, but just like on in bar graph form. But there's so much variation.
yeah. And,
Um, but yeah, like it's to the same point that you said in the last graph, right? Like these things are speaking to because we know something about the cluster or the life cycle stages that use these clusters. It tells us that the genes that we don't know what they do in these clusters could be doing something in the things that we do know that they do.
yeah.
Yeah.
So, like, previously, you've only seen little parts of the network. This one is seeing everything. And because of that, we're going to
Hmm.
see a lot of things that we don't know. And that's going to be
Hmm.
aspects for future research.
Yeah.
Uh,
A lot
a
of
lot
future
of future
research.
research. Uh,
Years and years. Decades and decades, maybe.
I mean, yeah. I mean, you think that, but then with techniques like single cell RNA, and it could be much faster than what we expect. And the way they've opened
Yeah,
this
well, I
up,
mean, this it opened it up because it allows us to ask the questions, but people still have to go and follow up on them,
right?
right? And like that takes time and research to get to drive down to the mechanism.
True.
Like this is only in some ways this is the I always found that these are best for hypotheses. Not all of them will be followed up on, right?
Mm,
Like they will in general, like when people drive to mechanism, those people are concerned about finding new insights from those mechanisms. Yeah, I don't know. It's kind of weird to think how research goes, but I
yeah.
think still decades from this sort of work.
Okay. Yeah. All right. All right. Care,
It's
care
just
to
silly
wager
fun. Okay.
$100 about whether malaria will be cured in a decade?
Yeah. Okay,
Okay.
so in Figure S13, they're setting up because they want to merge their
Hmm.
data sets. They're using a certain type of tool to show like how well these two data sets map onto each other. So from the falciparum, right?
Yeah. So they're
Hmm.
referring to falciparum as PF and Burgeri as PB. Um,
Hmm.
and looking at areas of similarity and dissimilarity. So we've got like uh, the kind of basic map of the atlas, their tops,
Hmm.
and also like this in yellow is stuff that is very similar, and in blue is stuff that's dissimilar.
Yeah. So there's a little cluster of dissimilarity inside of the sporozoite phase. And again, one of the things that they say here is that this is also data that isn't really present inside of the in the Burger Eye data set because they
Mm,
didn't dissect mosquitoes so in detail. um,
right.
but in general, the cell stages from each different data set map onto the other data set quite well, and so from this they're going to merge their data sets to try to get some new insights.
Right. So that brings us to ABC, which again I think it shows that overlap.
Yep, it's
Again,
like they're running figure one over again, but now
yeah,
with a merged data set.
yeah, and you can see like in P so in falciparum you can see that there's like some various, so these are like for divergent genes essentially. They've screened out all the genes that are similar and they're looking for the genes that are dissimilar. Right? Am I interpreting that
No, this
right?
is just these are the
Oh,
full transcriptomes.
oh, right.
Every
Yes.
dot is a
Apologies.
transcriptome, and they're merging them together.
Right. Oh, yeah.
And
I understand. Yeah.
we'll see. I think we'll see how they use it in later graphs, but this is just a recapitulation of figure
Mhm.
one, in that, but now they have two data sets sitting together.
Right, and basically this C1 cluster seems to be very dominated by falciparum. So
Yes, and so they're going to ignore that because they want to find, I guess they want to find genes that are conserved
mm,
between the two, right? They sort of hinted that in some of the supplementary because because they were encouraged, I guess, by seeing that they could get some gene cluster information. They're going to rerun clusters on here, the k nearest neighbors clusters, and
mm.
then try to say something about common functions or dissimilar functions.
Right, which brings us to 2DE, where they focus in on marker genes, I think. So
Yeah,
yeah.
so if they find things that are highly expressed in both these clusters, these mean that these are conserved between PF and PB. These are the common gene programs, if you will, or functions, yeah, between these two parasites.
Yeah, I think that's most. I, yeah,
Yeah, I think
that basically
that's good.
sums it
I
up.
don't really have.
Uh,
Yeah. Because at the end of the day they don't have anything to say really about those genes unless like we're not steeped in the malaria literature like as
mm,
well as others, but like presumably, right, like you would look at these types of genes, and if there is known function between the two, you'd say plasmodium, right? This might make a good vaccine target. I think that that's maybe one of the things that
um,
you could use this information for.
I mean, I guess these are like just benchmarks that everyone in the field might be familiar with. So, I mean, I looked these up. The SLARP, which is a plasmodium protein that's expressed in sporozoites and in liver stages.
Hmm.
CAP380 is an oocyst specific marker. Um Conserved plasmid protein is also known as conserved protein of unknown function, but it's present in the cytoplasm of both asexual and sexual stage parasites. So I think that that should be expressed a lot.
Yeah. So for the ones that they serve as markers to say this experiment. But for
Yeah.
the ones that you don't know, right, it's saying that these are things that are conserved between the two species and associate with a specific life stage, right? So it is giving us, again, this is one of the ways that you can come to a hypothesis about what some unknown gene does.
Yeah, I mean, and as you said, we aren't like insiders to this field. Not really had much training in Plasmodium since like my undergraduate degree.
Yeah. Like
Uh,
I don't know any of the like gene names that are coming up here.
so yeah, we are basically learning along with you at home with this. So um, yeah, next first slide is the best case. Sorry. Oh,
Oh yeah,
some.
so they pull out so again, like I what I felt through this beginning part of the paper is they're really trying to say why why is this analysis useful?
Mm,
Right? We collected the data. We know the data is looking at cell types that we haven't looked at before. We know that that is useful data. Why is this analysis useful? Right? Like why bother clustering, right, using this k nearest neighbors?
mm.
And what I think that the conclusion that they come to, and through like all the other figures, in some ways figure six is a small microcosm of the whole point, right, is that because they see clear separation between, so like I think it's for F and G really, because they see this clear separation where certain genes are turned on in a cluster and certain genes are turned off, and these genes we don't know what they do, right, but we do know what other genes do in those clusters, right? So that's speaking towards D. There
Mm.
are other things that we do know in those clusters, and what this data set is giving us is that insight that we could relate the things that we do know to the things that we don't know to help us ask a more specific question about the things that we don't know, where we have an educated guess, right? We actually have a little bit of knowledge to say that this is the right question to ask. And it goes back to what I was saying before, that science only answers the questions that you ask. And so these types of studies, I think, are really good in broadening and giving us a little just like the smallest hint of the next question to ask, right? The next hypothesis to put forward. That and it needs to be followed up afterwards. Yeah.
Yeah. I have nothing to add, so I'm going to move us to. Uh, okay, right. So this is looking at genes look unique to Plasmodium falciparum and not Burgeri. So
Mm-hmm.
yeah, this is quite useful for looking at those vaccine candidates or looking for things that might be interesting for host specificity. So Burgeri
Mm-hmm.
infects mice, falciparum infects humans. So this can tell us a bit about how the malaria reacts to the human immune system in a way that the Burgeri doesn't.
Yep. In some ways, you can think of this as the zoomed in version of those graphs with all the black in them, right? That showed us
Hmm.
conserved genes. This is the zoomed in version of that. Now, here are the genes, right, that actually are like in one but not in the other. And
Yeah.
that might be interesting, right? They're in specific clusters that assign them to a life cycle stage.
Yes. Indeed.
Let's trot
Yep.
along.
Figure S14, looking at the asexual stage data, uh, and
Same analysis, just in the asexual
yep.
stage, shows that there are like certain genes that are made in one but not in the other. And then they have a funny little control, D, E, and
Hmm.
F, where they look at the difference between the two strains of PF that they used.
Right.
And they show that there isn't that much difference between the two. And somehow that supports the fact that intraspecies variation isn't what's driving the phenomena. I don't buy it, but
Yeah.
like, um,
I got kind of confused about.
I put a question mark here.
Yeah.
Yeah, because I was like, I don't know how that
I
makes
mean,
sense, but
I
that's what
think
they
the
say.
idea of using those two strains is so that you could have that kind of say, oh, look at the things that you can decide by, because I think one of them is a laboratory adapted strain and one of them is more of a wild strain, and the idea, and I
Mm-hmm.
think you're right there.
Mm-hmm.
It doesn't necessarily like say anything about the geographic variation as much as it say if you say picked one from Southeast Asia, Sub-Saharan Africa, even if you've got like kind of a library strain from the 1920s somehow, uh, and see like whether that captures the variation. But
Well, it's like in ABC, they're saying PF
right.
versus PB, right? And so like that is like okay, and then so DEF is some sort of control on that to say, oh, but in closely related things, they don't have this difference. If they did have the difference, then you could imagine, okay, the maybe it's about the magnitude, right?
Mm.
Because there's some sort of like sensitivity metric that is being applied filter onto all of this stuff. And so maybe it's saying that like if they did see a difference in DEF, it would mean that the differences they say in ABC may not be so significant, right?
Right.
Because if those differences are also on things that basically are not different, then those differences don't say anything about the biology. But because they don't appear there, then they are somehow biologically
Yeah.
significant. That's what they're trying to say. I just think it's a weird way to show it.
I think it
Yeah.
does like speak to like kind of the robustness of their atlas because what they want to do is have malaria researchers being able to refer to that. And if there is a lot of intraspecies diversity, then that would harm the applicability of this. So I think that's
Yes, exactly. Okay, yeah, very. I'm on board. Yeah, I agree. That's a really good way of phrasing the utility of that control.
yeah, because I mean I know that in like my field there's a lot of like issues because the type strain was just one very tiny like reflected one aspect of it, but there's all this other diversity in the bacteria that we weren't really paying attention to that has become more
Hmm.
important to look at. And I
Hmm.
think that I mean that and now that we've got these big kind of scale technologies that we can look at things in such a big way that's changed the way we've kind of thought about type strains and diversity in all sorts
Hmm.
of
Yeah,
species.
in some ways, what we're seeing in DEF, what I'm hearing, my revised understanding now is saying DEF, it's making a statement to say we're not being overly blinded by the variation here. The types of variants that we're seeing, they have significance. They're not yeah, they're
Yeah,
not just noise that we got overly fixated on.
because I mean that was a problem in the early era of genomics where you'd find a gene that is associated with lots of things because you didn't have enough controls or you didn't kind of get a full picture of the diversity of expression of that gene.
Hmm.
So I think that now people are a lot more wise to that And so they're like, okay, we need to demonstrate that it isn't just like, say, because we caught this one specific weird strain. It's going to be applicable to lots of different things.
Yeah. 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 Very good point. Very good point.
So figure S15, another thing that they wanted to make sure about reproducibility. Uh,
Yeah. And this is because, as we said before, this right data that they have is like crazy detailed, right? They did all this crazy dissection, so different days of dissection of the parts of the mosquito. So this data set, they're going to dive in much deeper in the later figures, and they want to make sure that when they look at differences in this data set, it's not driven by the fact that they dissected multiple mosquitoes on different days.
yeah,
And that would drive the variation that we
yeah,
see.
and even if it did, I think it's important for a study like this, which is creating an atlas for a resource for other researchers to use, to be as transparent as possible about the limitations and about the kind of variations that might be in the
Mm-hmm.
data, because people might be using this for years
Absolutely.
to come. So you have to make sure that they have it out there, because I know that people won't read it, but it's important that they do and they understand
Well, I mean,
this.
if you're in the field, you're hopefully you're reading it, right? If
Yeah,
you're doing a study that's based on this, then hopefully you're reading it. But yeah, it's for us, it's excess detail that it was just good to know that they did it.
yeah. Um, so now this is focusing on the sporozoites a lot more.
Mm-hmm.
So looking at the
And
trajectory.
they subset them out, and they do the same UMAP cluster analysis and pseudotime,
Yep.
giving it that developmental trajectory. What's really cool is they also bring in another data set where they had two different types of sporozoites,
Hmm.
ones that were from the oocyst and ones that were from the salivary gland, and they just did bulk RNA-seq on them. And so they can compare the bulk RNA-seq to the single-cell RNA-seq and say that only 20% of the genes that were identified in those in previous studies were or sorry that were previously identified in that differential studies are in this data set. So that means there's a whole bunch of unknown stuff that we're picking at resolution.
Right. Yeah. Yeah. So that brings us to figure 3DE, which is progression of expression. Sorry. Uh, yeah. These later
Yes.
ones, they're very data. My brain's melting a little bit, but
Okay, yeah. So I guess the best way to start summarizing this last thing, because again, it's a story about sporozoites,
mm,
right? So they subset out the sporozoite data. They showed that what's really interesting is that in previous bulk methods they weren't able to find anything. So they zoom in and they make the funny clusters again, the G clusters, and they give them the top go terms, and they put them on the pseudotime graph, right? And they look at how those gene expression profiles change over time, and it gives them a sense that because they we're not in the malaria field. I have
right.
to say it again. We're not in the malaria field, but when people in the malaria field read this, they say, "Oh, like that gene that's responsible for doing these types of things," or like, "Oh yeah, I read a paper about that. That does this sort of function." And to see those genes represented on a timeline
Hmm.
gives researchers the confidence to know that these clusters are significant in that they're showing things that we already know, that we know these genes are supposed to go up and go down in these patterns because it makes sense in the biology. And once again, the whole point of this paper basically is to say they show us that this data set has things that we know, but it also has things that we don't know.
Mhm.
So the fact that we're being confirmed, the things that we know are being confirmed, gives us confidence to say that the things that we don't know might be viable hypotheses to now test, right, in ways that have the context from things that we've seen before. I feel like you're broken. This is like
Yeah.
I think I've said that several times, but that is all this paper is. It's just several stories. It's the big data set, and then they started breaking up in different ways stories about analyses of the data that for people in the malaria you should see confirmation of things that you already know, but then also hints about what you can ask next.
Yeah. Uh, so that brings us to the next figure, which is focusing in on two proteins, I believe, MSP1
Yes.
and P41, which encode
Hmm.
merozoa surface proteins that have never really been seen in the mosquito stage, so people didn't really think they played a role in the mosquito stages. And
Hmm.
some haven't really been elucidated, so no one knows what they do. But in this study, they find that there are some in the mosquito stages. So,
Yes.
so this,
And they
yeah,
even go and follow up in G. They stain a mosquito, right? Gut, and they find, oh, yeah, that MSP is being made at this specific stage where the sporozoite is bursting out from the oocyst. And that is interesting because that same MSP gene is used when merozoites burst out from the red blood cells. So I think this is maybe the example now of that broken record thing that I've been
yeah,
saying, a more concrete example of my broken record speech to say that here's where you're zoomed in and you're saying, we didn't know that you could guess that it would have the same function because it has to move through a barrier, but you wouldn't be able to guess it out of thin air. But
mm,
this data set, because it asked like an unbiased question, right? A hypothesis free type
mm.
of study. Um, it it allows us now to find connections that we wouldn't have found otherwise, or we may have found otherwise, but we've taken a lot of trial and error to get there.
Yeah, and I think this brings us to the fancy video portion of the that. Oh God,
Yes, they want
I'll
to
be
show off
so
their
okay.
oh,
Right.
it says module not found.
Oh my gosh. Okay. Well,
It all it is it's this image, this three colored image, that when people do, we haven't really talked about confocal that much, but when people do microscopy these days, they're able to filter out only the in focus light from a very specific point in the depth of a sample. And so what they did here is that they moved the focus through the depth of the sample. So it might be like a few microns. Oh yeah, here we go. It's
here we
only a
go.
few microns, but you know, these things are small. So like a few microns gives you a lot of detail. And they essentially do an optical like slice. I guess they actually call
Yeah,
it
an
that, right? They
optical.
optically slice through. Yeah,
Yeah,
construct that, and you get this awesome 3D image. This is a moment in the sporozoite development, right? It's bursting out from these eggs after the eggs got planted on the outside of the mosquito, and it's stained for this MSP in red, which
yeah,
is a protein that they just never looked for it in this scenario before.
right.
And we already have data, we have studies that show us what it does in a different part of the life cycle. So it's very exciting, I think. Shows you the promise of such an atlas to be able to, to show us stuff like this.
Yeah. So back to the back to the slideshow, and now that's figure 16. This is also looking at the proteomes and how those change
Yeah,
over
well,
time.
this is proteomes. We haven't looked at this, but yeah,
Yeah.
this is once again the pleasures of omics research is that there's all these other ohms out there that you can compare to your ohm. This is proteins that are actually made. And what's interesting is that there's a disconnect here,
Mm.
right? Not all the proteins, not all the expression, the transcripts are being made. And that's fascinating because there's a whole other part of biology that we also haven't talked about, but stuff that is being regulated on the translational side.
Right. Yeah. We don't. There's all the stuff that RNA can do that doesn't necessarily require it to be translated into a protein. And so
Hmm.
the question is, what is it doing? And now we have that kind of by comparing this data with the data that's been acquired here, you can get more an idea of that knowledge, and or even maybe pick out like which proteins might not be being detected in the proteome screening, or if there's something else interesting in regulatory that we might not.
Absolutely.
Yeah,
So yeah, another interesting bone that they've thrown us.
yep. There's lots
um,
because yeah, there's lots of stuff they've
Continuing on the theme of interesting bones.
yeah, looking at sporozoic genes that are highly variable in expression. So the
Hmm.
this is kind of because again throwing another massive thing at us because the idea is, I think,
this is sort of taking a step away from the cluster now,
mm,
right? And saying that they had these co-expression clusters, but they also find just genes in general that in certain single cells are widely expressed high or low. And that's weird, and they don't correlate with each other.
right,
So it's not that all these cells are in the same boat. They're in different stages of life, and they're using genes. They're using these genes, and I think the authors hypothesize that these could be because this is single cell data, right? These could be cells that find themselves in a certain scenario where they have to course correct
right,
in some way. Yeah.
yeah,
Which is really fascinating. It speaks to like what exactly is the core function, right? There's a core program that's being executed, but then in certain circumstances, right, the core program has to be realigned, right? You have to push back into the core program because maybe the environment changed drastically, right? Because remember, these are tiny single cell organisms that are swimming in relatively large
yeah.
organisms, and they may find themselves in a scenario, maybe they get a squared off with an immune cell or something, and then they have to do something in order to survive that encounter, and this could be the signal of that. I think that's what the authors want to
Yeah,
suggest.
and I mean it could also be almost an artifact of the fact of their times framing, because if there's a gene that is on for maybe 5 minutes during the entire cycle of the Plasmodium, then you might only capture it on like very rare occasions.
Yes,
So
and then there's not enough events to actually form a distinct cluster with those things, and so then they would not get picked up as like its own program.
so yeah,
That is a very good point.
there are like various alternative hypotheses and that's why this is actually really fascinating, because trying to find out what these
Mm,
kind of highly variable genes are doing could be really interesting. Um,
yes, yes.
I mean, if both, I mean, I guess in the first hypothesis, the one they proposed, that's even more interesting, because the idea that these things are smart, they're reacting to individual situations in a way that we don't know, and that could
Hmm.
be quite interesting, because the same thing could be happening in the human side of the infection as well, that we're not capturing it because we.
Yes,
So
yes.
that is also very fascinating. Um, yeah. So moving on to uh, so and back to the AP2 transcription factors.
Yep. More I think these are like another set of transcription factors. So many transcription factors. And in this case, yeah, they're seeing in the pattern. It's really this is again very
Mm,
much in conversation with the field that we're not in. So like a lot of it went over my head, but it sounds like what they were trying to say here is that there's a lot of transcription factors. They know they're important,
mm,
and there's a theory that these transcription factors control each other, and seeing them in this particular data set, they're in a very similar cluster, and that gives them some support for that hypothesis that they're just like, yeah, really useful. They have dependencies on each other.
yeah, and I think that's a really tantalizing thing, because we found that for bacteria, there are some like unique clusters that you can target for like say quorum sensing that can like trigger an entire like kind of cluster upon cluster to change bacterial behavior, and this is like doing that opportunity of reading the mind of what is the Plasmodium thinking when it's in the body, and these transcription
Yeah.
factor networks can help like pull that apart.
Yes, absolutely.
So
I agree.
so that's
I have no
exciting.
more to add. I have no more to add.
That's fine. I pulled the same stuff that's done on you for most of these slides. So yeah
uh, we're getting to our final figure,
yeah
um,
th-
which is actually like another little experiment that they pull out of their back pocket. And actually, reading this reading this paper just made me appreciate like where these like the journey these slides go on to get to where they need to go. Um, and so you imagine that they have to once they get injected into the once the mosquito injects them into the from the saliva, they find the bloodstream.
yeah
um, and so like they could just be in like the intracellular space, and they're just moving through tissue and basement membrane and stuff like that. And I love this reading. The one sentence that I read saying that they may invade fibroblasts several times in an attempt to search right for the
Right.
bloodstream. And that's crazy to me. So like these tiny little single cells are into other cells and like poking into other areas. This period of their life, they're also exposed to the immune system in a way because they're
Yeah.
living outside of cells. So the authors want to zoom in and figure out what's going on in these very specific stages. And so what they do is they make an artificial environment that tries to mimic that, uh, being that they have the salivary sporidium, and then they have a permeable membrane that's covered in fibroblasts.
Mm.
And they want to see whether or not the addition of those fibroblasts would trigger something in those sporozoites to go undergo some other developmental regime.
Right, yeah, this is interesting because again, sporozoites, pulling back to the beginning, they're the target of most vaccines because this is their vulnerable stage. This is, and yeah,
Mm-hmm.
the way they migrate, it can be narrated by David Attenborough. It'd be like, oh, here, yes, we have the sporozoites moving towards the dermal to find their target, but oh, they can't find it. And
Yeah, no, that's that. Reading that sentence, I was just like, it really felt like, wow, like this is their world. Their world is just cells, and they're just swimming along trying to find the right place to be.
yeah, and it's fascinating. Also, like it's also really important for research to be able to model like how the skin works because this is
Mm,
quite challenging work to try and figure out because
yes.
at the moment we've seen all the aspects of the mosquito's like cycle apart from the last one where it bites a human and injects the sporozoites in because we've seen sporozoites like going through all those stages of development, but how does that change when they enter a human and where
Hmm.
they actually have to migrate towards their target, which is the liver.
Yes. And but I guess kind of surprisingly, interestingly, their whole experimental design really doesn't affect anything.
Mm.
These cells change just on their own, it seems.
Well, based on an internal clock, but also like
Mhm.
to be fair, they don't really have any comparisons to the natural like situation as far as I could tell. I mean,
Ah,
I
yes, very good point.
so I mean, well, at least not on this set of the experiments. I mean, but I guess they might have some comparisons to other people's studies if you take like blood. But again, it's very hard to sample these in a native situation. You have to find someone who's a malaria patient who's willing to be subjected to an invasive experiment, and also you have to capture these sporozoites very earliest stage of infection. So this is
Well,
before
this
someone's
this
been
this would be something that you could probably do with mice, right? You might be
yeah
able to capture this stage in a mouse, but yet probably not in a human because it's like before it's when the sporozoid is injected into your body, but before it gets into your blood. So what sort of biopsy would someone consent to get like that enough material to do a study?
yeah
That
you
that sounds
would
tough.
not be able to find the people because if they're naturally infected, they wouldn't know it until you see the symptoms. So this is very
Hmm.
so you'd have to deliberately infect someone with malaria in order to find
But
this
in
out.
but in malaria, they do actually deliberately infect people to do their
Right.
studies. Yeah, I know that yeah, it's done there
Okay.
because there are a lot of therapeutics.
True. That's fair enough. Yeah.
Okay,
But in
anyways,
this,
so
yeah.
that's what the authors are trying to do here. They're trying to find out this elusive stage. They find that doesn't really matter if they have cells on their experimental design. The sporozoites are just changing over time.
Right. So it's almost like there is an internal clock in there that causes them to
Hmm.
change. Um,
Hmm.
I mean, like,
And they also have some bulk expression data from Plasmodium Vivax, yet another malaria parasite.
yep.
And it seems like it kind of correlates, right? That they see a shift too when in Vivax. So I guess they did do an experiment somewhere
Yeah.
along the lines. Yeah.
Uh, and 4DE the so again look this is an actually odd sort of thing because you'd think the sporozoites would have lots of cell traversal genes in order to make their way through the body, but it looks like they're
Hmm.
being downregulated in this experiment. Um,
Yes. Yes. Well, they get downregulated, but then other stuff gets upregulated.
right. So stuff
Yeah,
that
and the
sorry.
oh, I
Oh,
was gonna say
sorry.
the thing that's getting upregulated is essentially the secretion system of
Hmm.
Plasmodium. So that's interesting from the point of view. It's like they don't need to move so much anymore, or very quickly they don't have to move, and then they start secreting things. I don't know.
Yeah, because I feels like the PTEX complex is this kind of arsenal that it gets ready in order for the point where it gets ingested by a cell and then it releases a bunch of stuff.
Hmm. Yep. Yeah, like good for the intracellular living part of their life cycle.
Yeah, and I think another thing they point out is that some of this like was seen to be like being readied up inside the mosquito salivary glands. So I mean,
Hmm. Yeah.
the interesting thought I had was that if we waited a bit longer in the mosquito salivary and would it have activated the salivary glands if we'd waited a bit longer? And
I really like that thought. Yeah. Is it yeah, is there maybe a salivary gland that inhibits this transition? Right? Like what about thinking about environments that inhibit gene transitions instead of environments that induce, right? Maybe that's the one of the conceptual missing conceptually missing from this model that they've proposed.
well, yeah, there's always limitations, and but yeah, this is going more into that PTEX 150 where they're seeing like these. So these are individual sporozoites that they're looking at and looking at the
Mhm.
activation of that gene within them, and so you can see these little granules appearing. Um,
And they quantify it in G.
yeah,
So again, this is one of those things that you wouldn't expect this. Like this is not a hypothesis that someone would have drawn out of thin air potentially. But after finding the signal for it in this data set, and they follow up and they see it in immunofluorescence, it sort of opens up a very viable hypothesis, right? Why does this happen, right? When does it happen exactly, right? How does it happen in
yeah. I mean,
yeah.
this could be really interesting. And I think it's also while it's important to take into account the model. I think that can still tell us a lot. I mean, this study is amazing in the amount of data it produces. So I think this is the last figure from the paper that we're looking at. But
Yeah.
I'm going to pull up the other big thing from this paper, which is the Malaria Cell Atlas website, which you can visit at
Hmm.
sanger.ac.uk/tool/mca/mca. Oh
Ooh,
no, my.
that's my fire alarm.
Okay, all right. Well, Terry, we can. Is it okay? I mean, we can stop the broadcast if you're on fire.
No, it's fine. I think it's just the kitchen. I think my partner's cooking something.
Okay, all right. Okay, but this is just a YouTube channel. You can run out of the building. I'll let me deal, and I can deal with the rest. Okay. Um, but yeah, no. Anyway, so yeah, this malaria cell atlas. So you can actually at home you can explore what they found in here based on this paper, and you can even go like so
Hmm.
you can look at focus on different lifestyles, and you can also look at genes. So you can pick a favorite gene and decide to look at it. I don't know what
My
any of these numbers
gosh.
mean, but you can see when it's expressed.
I
I
mean, some
mean,
of these are the unknown genes, right? Some of these are the unannotated unknown genes.
yeah.
And yeah, like
So
this is
uokinete. So I think this is in like so gametocytes and in the uokinete. So who knows what that does?
yeah.
But it's quite and pick another random gene. And what else can we find? Oh look, this one's expressed in this stage as well. So you
Yes.
can go wild and drive yourself slightly insane by going through this and exploring stuff. But it's fascinating. They've got this resource that's available for all of these research for everybody to use and look at.
So like even if you're not particularly like computationally inclined, you could go in here and sort of confirm little things, little hunches maybe that you read. You can go in and look at the data set. And of course, the data I believe is probably accessible through some sort of API. So if you are computationally inclined, right, you can pull that down and run some of these analyses yourself.
Yeah, and they've got pretty much most of the analysis they use in this paper. So, yeah, it's meant to be a living resource as well. So they've published multiple papers that are fed into this website. So you've got Plasmodium bergi and Plasmodium nosy, which
Hmm.
yeah. So the bergi is quite fun because it's 3D. Uh,
Ooh.
yeah.
Huh.
So
Oh, that's because they were using PCOA instead of UMAP.
Yeah, so
Yeah, so
this has
PCOA
too
is like
many.
an older way of dimensional reduction, and so harder to it's harder to collapse things into 2 dimensions and still get good resolution. So they did 3 dimensions.
So yeah, you can see, even look at how different clustering affects. So PCA, you can see like, oh, there's this big like kind of loop that loops around, and then you can see how UMAT deals with that loop, which is to separate it off from everywhere else.
Yeah, yeah. Yeah, so I mean, this is just, it's kind of nice. We could probably talk forever about the differences in the algorithms as well,
Yeah,
because they sort of drive, I mean, it drives the type of analysis and the way that we see things. I think most people are just happy having an upgraded version that you don't have to use a third dimension and still get good separation.
right. Yeah. So
Yeah, I
yeah,
mean, all that to say, this paper I think was particularly challenging for us to read because it was in such close conversation with pre-established malaria literature. So they would drop, I mean, anyone has read it with us, like they would drop these sentences like, as you know, this gene like that's upregulated here, and like the little citation to another paper. I'm like, I'm not reading a whole other paper just to understand the sentence, right? Like, I'm just gonna trust that the field knows what it's talking about, that it's speaking in conversation, and what am I gonna get out of it, right? And I think what I've gotten out of this paper is, first of all, I've got a huge appreciation of the complexity of the malaria cell life cycle and the way that
yeah,
it goes through all these different stages. But that broken record thing that I'd been saying through the whole episode, it's a it's a case study in a way of how does single cell data get used to drive hypotheses and to drive discovery, right? Because they did such a good job about just describing their analyses that even not coming from their field, I could say, oh, well, like I see how this tool is being used
mm,
in a field, right? And so if I was in a field that I understand more about, like if I knew Staph aureus, I know more Staph aureus genes, right? I can now understand what would single cell sequencing in a Staph aureus infection tell me, right? How could
right.
that drive some understanding, right, that I wouldn't have previously? How would I leverage the knowledge I already had to find genes of unknown function and then assign them function? Because like that's real hard stuff to do, and these types of data sets are very well positioned to give us that information.
Yeah, I agree completely with that. I think that, I mean, this paper was also, I definitely agree that this was very challenging. That I've learned more about Plasmodium in the past week or so trying to learn about this than I did falling asleep in my lectures trying to learn about it. So, uh, I mean, it's given to me a lot of appreciation for also the kind of complexities of the life cycle, and also the complexities of fighting the disease like malaria, and also how unique and interesting this
Yes.
pathogen is. That, I mean, again, like I'm not used to dealing with much simpler cells, and this has got all these degrees of complexity, and and
Yeah.
that comes out in the data analysis of this paper. And yeah, I did feel like I was like chipping off a small slice of a much bigger iceberg.
Hmm.
And yeah, like just like you said, I'd be like, oh well, they refer to another paper, and I'd look at the title and be like, I just don't have the time to read all this, so I'm gonna have to just trust. So there is going to be a slight downgrade in the level of critical like analysis that we do, basically
Yeah.
because it is outside of our field. So I'm not going to be able to pick. We're not going to be able to nitpick.
It's two things that are outside our field. One, the biological relevance of this data. Two, like the actual computational analysis. Like
Yeah,
I think we know enough. I mean, I feel like I know enough to like understand what they're doing, but to understand if what they did was right, that's like something that I don't think I was bringing to the table when we read this paper.
yeah. I think another thing. Yeah, this paper does highlight the need for data analysis skills, like how it has changed so much over the past decade. There is so much like more
Hmm.
nuance, so many more tools out there that, I mean, because UMAP is basically an AI, right? That passes through things. It's machine learning.
It's a machine learning.
Yeah,
Yeah, it's a machine learning algorithm, I believe.
yeah,
It's
and
a classifier.
yeah. So I do believe that that's going to be quite important.
Ooh, no, actually, I would not say it's no, it's not. I not. Because
Okay, right.
the advantage of UMAP is that it is based in a known like mathematical theorem. It's actually it's a type of like graph theory.
Right.
Yeah.
Okay.
It's not a machine learning. Yeah, it's not. It's
Shows
a
I need to learn more about
yeah,
this.
yeah,
Yeah.
yeah, yeah.
But yeah, I mean the fact that our data analysis strategies are becoming very kind of
They're very
very nuanced.
algorithm focused.
Yeah, very
Yeah, we have
algorithm
we have to
focused.
sort of understand these algorithms and the interplay of them together.
Yeah, and it's interesting because like I went to biology to escape mathematics, and now like several years later, it's found me and it's coming for me. So I need to like buck up and try to sit down and learn these things now because it is becoming a part of our like job to understand how these work.
Absolutely. Absolutely. Okay, well, if there's nothing more that we have to add, then join us next week for our news week, where we're going to survey a bunch of different papers to try to find something that we're going to cover, just like we did today, in the following week.
And we want to remind everyone that while we're very enthusiastic about microbiology and somewhat qualified, it's possible we didn't get everything right. So science is about thinking critically and asking the right questions. And if you have any questions or corrections, please let us know in the comments.
I 100% agree. You can reach us out to us over Twitter with the hashtag #MicroTWGC. We both believe that peer review is a process and that we can all participate in it. So we hope that you've had a really and stumble through this paper. If you think you have something to add or found something unclear, just let us know.
It has been a pleasure chatting with you, Danny.
Same here, Fuzz.
Tune in next week for more microbiology content.